This video will discuss the Phytoplankton Lab for the shellfish course. I will be showing you some different types of algae and how to actually take a sample to look at under the microscope to get your cell counts. So the first algae that I have to tell you about is Isochrysis galbana, which is the T-iso. It's a flagellate, so it's motile. It's oval in shape. It grows between 17 and 30 degrees Celsius. As you can tell, it's golden brown in color and it's very small, usually around 4.8 microns by 4.6 microns. Under the microscope, it would look like this picture right here. The next algae would be your Tetraselmis, and it's a Tetraselmis species. It's green in color. It also is a flagellate. It varies greatly in cell size and cell shape. It can be round, ovoid, elliptical, compressed. It usually grows between 10 microns long by 14 microns wide, and it grows between 19 and 21 degrees Celsius at 20 to 35 parts per thousand. The next algae that we will talk about is the Pavlova species. This also is a flagellate, so it's motile. It's oval in shape. It grows between 11 and 26 degrees Celsius. It's golden brown in color, and again, it's very small, 3 to 6 microns. All of these algae are being grown to feed to oysters, so they have to be pretty small. So the Pavlova species would look like this underneath the microscope, like that. So the last species that I want to tell you about is the Catastrophes Mülleri. It's also called a Shagra. This is a diatom, actually. It's different from the rest. It's not motile. It has a silica shell and is rectangular in shape. It grows between 15 and 30 degrees Celsius and between 20 and 35 parts per thousand salinity. It's yellow-brown in color and 4 to 9 microns by 4 to 10 microns. And it will look like this underneath your microscope. So it looks like rectangular shapes. So what we normally do then, when we get all of our algae, we take a sample and we use a special slide called a hemocytometer to look at your algae through a microscope. So you just put your sample on the little wells right here and here, and you use this cover slip to put over the sample. Then you're able to look at it through the microscope. When you look at it through the microscope, you're going to see a grid like this. So you're going to see your grid like this, and what you need to count are the cells in the 4 corners. So this corner right here, you count those cells, those, and those. And once you get your count, you have to, there's a calculation that you do that's in your lab manual. So now I will take some samples of all of these, and I will provide you with the cell counts so that you can finish up your lab.